Thank you very much, Helmut. I'd also like to thank the organisers, and Josiane in particular, for organising the meeting and for inviting me to Paris. I haven't been here for 20 years, so it's very nice to be back here again. What I'm going to do is to outline some of the work that's been going on with my lab and with collaborators, which point out some of the interesting aspects of mitochondrial function and how we can actually intervene both to modify mitochondria and to design probes to tell us what's actually happening inside mitochondria in vivo. Now, therefore, that's the overall story I'd like to get to you, is the idea that targeted molecules might be able to intervene and tell us what's happening with mitochondria. To do this, I'm going to go through one little story that illustrates some of those examples of how you can use these probes to play around with mitochondrial function. So, I'm going to focus, this is a heart. It's an exquisitely organized physiological machine that's been studied for centuries by scientists and physicians, and we understand it in exquisite detail. But I'm afraid, to me, a heart is just a big bag of mitochondria. So anyone who really knows about the heart should switch off now, because to me, all the heart is a bunch of mitochondria doing their job, and if the mitochondria go wrong, the heart goes wrong. So I'm sure it's a bit more complicated than that, really, but to me, that's how I see it. So what I'm going to talk about is some of the aspects of a heart attack. Now, in a heart attack, what happens is the blood flow stops to the heart, and then you enter a period of ischemia or anoxia, where the mitochondria are anoxic and are not actually supplied with oxygen. And then after a while, that goes, the blood is reperfused into the heart, and it's during that reperfusion that a lot of the damage occurs. You end up with a burst of reactive oxygen species, and the important bit is that you get a burst of ROS production in the mitochondria, which leads on to things like the mitochondrial permeability transition pore and cell death, and then a whole bunch of boring downstream things like cell death and inflammation and death of the patient. But the important stuff, I think you'll agree, is the actual few minutes where the mitochondria produce a burst of ROS early on during reperfusion. Now, how are we actually going to deal with these mitochondrial events? We want to be able to intervene in mitochondria selectively, and what I've been working away for the last decade or so is trying to understand, can we actually design drugs that go straight to mitochondria? And we're trying to develop a field which I've called mitochondrial pharmacology for obvious reasons, that we want to think about mitochondria as where we should be sending the drugs. We've done a whole range of different drugs, pharmacophores and bioactive molecules that we can target to mitochondria. And we've used this trick here, which is very straightforward. What we do is we tag the pharmacophore, a drug that we want to go to mitochondria here, with a lipophilic cation, a phosphonium here, a phosphorus group, which was first used by the Skuleshov group back in the 70s to measure mitochondrial membrane potential. And what we did was use this idea, which is not rocket science. You just take this molecule here, which has the weird property that it can go straight through phospholipid bilayers without requiring a protein carrier. And because of the positive charge, it tends to accumulate inside mitochondria, but it accumulates dramatically, maybe 1,000-fold, due to the Nernst equation that describes the uptake. So all you do then is just add your favorite molecule or probe here, and then this molecule will go in orally or intravenously or intraperitoneally into your favorite organ, into the cell, and into the mitochondria. And it can accumulate many thousand-folds inside mitochondria, and also the other thing is it can accumulate extremely rapidly. So if you do an intravenous injection of these molecules, they'll be in your heart mitochondria within a couple of minutes. So you can intervene acutely as well as chronically. Now, we've made many of these, some of them like MitoQ, a mitochondria-targeted antioxidant, have gone on to clinical trials. But we had the idea that if we want to intervene in heart attack, one, we need it to go in very quickly to the heart, because you want to intervene at a critical stage in the heart attack. And two, what do we know about the heart that's modified that might be protective? Is there anything, apart from just blocking oxidative damage, how can we actually intervene in the heart to protect it? So, those who do know really about the heart and have been working on it for a long time have come up with some indications that S-nitrosation or S-nitrosation of proteins in mitochondria inside the heart might be protective. This work is primarily correlative. In other words, if you set up situations that protect the heart, such as treating with nitrite or something called ischemic preconditioning, you end up with a bunch of proteins that are S-nitrosated or S-nitrosylated. What happens is that this is a protein, and it's got cysteine residues. Many of them are structural or in the active site or bind to iron sulfur centers, but some on the surface are protective or potentially regulatory. A very small proportion of those on the surface can actually be modified by things like glutathione. Well, many can be modified, but some of these modifications can actually be regulatory. A very small proportion, though. One of the modifications is this S-nitrosation, where NO metabolites, in slightly mysterious ways, generate an S-nitroso compound which can modify the activity or location or interactions of the protein. Then this is reversed and you end up with this way of modifying the activity. What seems to happen inside mitochondria under conditions that are cardioprotective is that you get a buildup of S-nitroso compound proteins there. It's a very small number. Now, the problem is that we don't know how they're protective, and we don't really know for sure the particular proteins of the ones that are modified which are protective. So we said, well, we can target mitochondria, and there's a hint that this modification might be protective in mitochondria against ischemia, reperfusion, injury, and a heart attack. So if we put those two together, let's make a molecule that can selectively induce this modification in mitochondria in the heart. So we made this molecule here a few years ago now, in 2009, called MitoSNO. So you've got the same targeting moiety here, the phosphonium, and here we have an S-nitrosating moiety based on the commonly used molecule in the NO field called SNAP. You have a protected SNO, and the notion is that this should go into mitochondria, and then it can transfer the nitric oxide, formally transferring the nitrosonium onto a protein thiol. So what will happen is this will go zooming into mitochondria, accumulate several thousandfold. It'll do a few things, but one of the things it will do is it will transfer the nitrosonium onto protein thiols, and we were wondering whether that would be protective during ischemic reperfusion injury. So this early work showed that we could actually induce this modification extensively inside mitochondria. Now, let's move this on to the in vivo situation and see, when we induce this, is it actually protective? What we do is use the LAD mouse model, and what you do here, it's a very good model of a heart attack. What happens is you expose the heart here, then you tie off the coronary artery so that you can induce for about half an hour anoxia or ischemia into a large area of the heart. This mimics what would happen if a blood clot blocked up your coronary artery. Then you'd have a pain in your chest, you'd go to the hospital, and they would probably put in a stent or induce restenosis in that artery to try and get the blood flow back. And you can mimic that here by just untying that. And then you get a burst of oxygenated blood going back into this anoxic tissue, and at that stage you get a burst of ROS production that leads on to much of the damage. The more damage you get, the more cell death, and even if you recover, the more damage to the heart, your longer term prognosis is poorer. So you'd really like to be able to prevent that damage during the reperfusion phase here. So what we want then is a drug that you can actually add at the reperfusion phase. Now, if you stoke up your heart with huge numbers of mitochondria targeted antioxidants like MitoQ, then they will protect the heart during this phase very nicely as well, and you get far less ischemic reperfusion injury. Problem is, people don't plan their heart attacks very well, so they don't know for sure when it's going to happen. And most of us will wake up after a heart attack in a hospital, and at that stage, what should have happened is that any drug would have to be added during the restenosis when you're unblocking the artery here. So you need something that can be added at that stage systemically and go to the mitochondria in the heart if it's to be effective. Now, we know that MitoSNO and these compounds, if you inject them systemically into the circulation by an intravenous injection, they go to the heart very rapidly within a couple of minutes. So we took that information and tried to see, could we protect the heart in this model? What we did here is you had ischemia for 30 minutes, and then we untied and reperfused, and then we followed the heart for another 2 hours. Very common model of a heart attack. But what we did here is we actually added the MitoSNO just before we untied it by a tail vein injection. So we're adding the MitoSNO systemically to the overall circulation, not to the heart, mimicking a clinically relevant situation. And we're adding it just the earliest time we could, just as we were about to reperfuse. So that would mimic what would happen in a hospital when you're restenosing an artery. And what you can see is here we measure the area that's damaged. And here you can see that in the control, about 30% of the area of heart that's damaged, indicated by this white area of dead tissue, is dead. But with MitoSNO, the area that's dead is far less. You really protect it here quite extensively. Control compound without the NO is not protective, and a nitric oxide donor that doesn't get to mitochondria is also not protective. So here we've got a very good indication that adding a amount here systemically is protective. And the doses we add, which is 100 nanograms per kg, very, very low dose, showing that we're getting exquisite uptake into the mitochondria as we hoped. So now that we've got protection, how does it actually work? Because if we want to understand what's happening, we need to know for sure what's the actual mechanism of protection here. So to look at that, the first thing we did was to see, well, would it be protective if we added it a bit later on or a bit earlier? And what happens is that if we add the MitoSNO 10 minutes later, it's not protective at all. So this is the same data as before protection, but added 10 minutes later, it's not protective. So in other words, it's only protective when added in a very narrow window around the reperfusion of the anoxic tissue. That's very interesting, as that tells us a special mechanism going on. It's not just loading up with protective stuff. So, and how is it protective? Well, the prejudice I'd start with would it be stopping cell death by blocking ROS production by mitochondria. Is that actually the case? Well, first off, we could show already there was far less necrotic cell death. We could also look at apoptotic cell death and the penumbra around the dead tissue, and you can see that ischemic reperfusion injury greatly increases apoptosis, and MitoSNO blocks that on these two measures of caspase activation. So yes, it's blocking cell death. Is it through preventing mitochondrial ROS production? Well, we actually developed separately a method by using mass spectrometry where we could actually measure the levels of hydrogen peroxide inside living tissues. This hadn't been done before, but it's quite important to measure the ROS levels, not the oxidative damage, the actual steady-state levels of hydrogen peroxide inside mitochondria in a living tissue. We did this by inventing a new probe called MitoB, which goes to mitochondria and has a boronic acid component that's selectively converted to a phenol. Then if after a few hours you can measure that ratio by mass spectrometry, you can measure femtomole levels of these probes. And then you can see for the first time inside the heart during ischemic reperfusion injury, you do get an increase in mitochondrial hydrogen peroxide production, which is prevented by MitoSNO. Then downstream of the production of ROS by mitochondria, then you also get an increase in protein carbonyls as a measure of oxidative damage, which is again prevented by MitoSNO. So all of these are consistent with the idea that MitoSNO is actually preventing mitochondrial ROS production during that critical window of ischemic reperfusion injury, during the reperfusion phase, and that's the intervention that then prevents the oxidative damage, cell death, and downstream sequelae of heart damage. So, can we understand more about how that's actually having its effects? Now, there are lots of ways that nitric oxide donors could intervene, but what we found was that it wasn't through any of the normal NO effects. I won't go through the data, but we could show there was no effect on hemodynamics. There was also no effect if we knocked out the normal NO signaling pathway by knocking out protein kinase G. So for those and many other reasons, we are as confident as we can be that the mechanism is due to S-nitrosation. In other words, it's nothing to do with free NO, it's to do with the S-nitrosation of protein thiols inside the tissue. So where is that S-nitrosation occurring? Now, in a method that Neil Hogg introduced a few years ago that's proven very useful, we can actually tag protein thiols that are S-nitrosated quite selectively. This is in the heart of the mice, and we can pull out the proteins and we can tag with a fluorescent probe the ones that contain an SO group, but leaving behind all the other protein thiols. If we do this this is all done where we're injecting 100 nanogram per kg of MitoSNO into the tail vein of a mouse undergoing ischemic reperfusion injury, and then we're isolating the heart and isolating the mitochondria, the cytosol, and the plasma. And what you can see is that MitoSNO S-nitrosates mitochondrial proteins, but it doesn't S-nitrosate cytosolic or plasma proteins, as you can see by these fluorescent tags here. That's important, and the reason for that is because it gets sucked into the cell, into the mitochondria, and accumulated so quickly at these very low doses that it leaves no imprint at all on proteins except those in the mitochondria, because it gets sucked into the mitochondria so quickly. In contrast, an untargeted NO donor has no effect on mitochondria, but labels the cytosol a bit and the plasma a bit because it doesn't get dragged into the mitochondria. If we try and identify then those proteins in the mitochondria that are modified, can we do that? Well, we tried extensive redox proteomic techniques, and we couldn't pick up any of those normal soluble proteins that we expected and we would see in vitro in extreme concentrations. That was interesting, suggesting there's a very selective subset of S-nitrosatable proteins in mitochondria that are responsible for this effect. The redox proteomic techniques we used wouldn't have picked up some of the mitochondrial complexes, so then we did blue native gels on the heart mitochondria from these animals. And again, with the same labeling, we could see that we were actually very selectively labeling complex 1 and complex 5. Complex 5 had no effect on the activity, so we think that's just a side effect. We don't think it has a functional consequence, but we're trying to confirm that. But complex 1, which is the entry point for electrons into the mitochondrial respiratory chain and a major source of reactive oxygen species, was very effectively S-nitrosated. So we think most of the effects we're seeing are due to complex 1 S-nitrosation. But of course, we want to go beyond that and understand a bit more about the mechanism of that interaction. So the first thing we did to see is, well, are we affecting the activity of complex 1? So we tried to do the same experiment, isolate complex 1 and measure its activity ex vivo. obviously, so the exposure was in the mouse undergoing ischemic reperfusion injury, but the measurement of the activity was in isolated mitochondria subsequently. So the interesting thing was, if we add MitoSNO to a normal heart and a normal mouse, MitoSNO has no effect on complex 1 activity. If we add it just on reperfusion, so we're adding it to an anoxic heart where we inject systemically and it goes into the anoxic heart, then we measure complex 1 5 minutes later, you get extensive decrease in complex 1 activity. Interestingly, we can, ex vivo, if we treat that with copper ascorbate, which selectively breaks down S-NO, we recover the activity, suggesting we get a decrease in activity due to the S-nitrosation of complex 1. If we leave that then half an hour during reperfusion when a lot of the damage will play out, what you can see is the complex 1 activity comes back up if we treat it with MitoSNO. So in other words, we were lowering the activity during reperfusion, and then if we leave it in the heart, it recovers over time and there's no further S-nitrosation, suggesting a reversible modification in the heart. Interestingly, if we didn't have MitoSNO, while initially the complex 1 activity was fine, over 30 minutes the accumulation of oxidative damage over that time lowered the activity. So in other words, complex 1 was protected by S-nitrosation by lowering its activity transiently, and then it came back over time. But if we didn't have it, complex 1 activity was initially high, presumably producing ROS, and then going down. But can we understand the mechanism by which this protection occurs? Well, to do that, we need to understand how we're actually modifying complex 1, which cysteines. There are many, many cysteine residues on complex 1. We hope there'll just be a few selective ones that we want to look at. So can we pull those out? Well, again, if we tag isolated complex 1 in a DIGE type process where we can tag it with a green and a red tag, and if we set this up in a certain way, we'll pick up normal iron sulfur proteins will just give a yellow fluorescence, but those that have been S-nitrosated will give a red fluorescence. And then we do a diagonal gel where we separate complex 1 by blue native, then we take it out and do a diagonal 2D gel to separate out the complex 1 complexes, or subunits, of which there are 45. And the red ones, which you can see down here, are the ones that are S-nitrosated. That's an artifact. And those 4, we can reproducibly show those are S-nitrosated. So we've got 4 candidate subunits within the 45 of complex 1 that are S-nitrosated by MitoSNO inside the mouse. So now that we've identified those, we want to now go on and identify which cysteine residues are actually modified and responsible in these complexes. To do that, we use a very simple mass spectrometric approach. Here's complex 1, and we imagine that we've got a mixture. Here we want to both identify the cysteine, but also vitally important is quantify the extent of the modification. Because what often you'll see is that the modification will often be found in generally post-translational modification. You'll find a modification, but unless we can quantify and show that it's a substantial modification, it may be biologically meaningless. What we do here is, if you've got a mixture of modified and unmodified complex 1, we first, with S-NO here, we block all the free thiols with the alkylating agent N-ethylmaleimide, the blue dot here. So we block all the thiols here. Now we treat the complex with copper ascorbate, and then we retreat it with NEM, but this time a deuterated or heavy version of NEM, so chemically identical, but it's got a higher mass. And so we've tagged the S-NO cysteine with this mass tag here. Then we digest these to get the trypsin, with trypsin to get the peptides, separate an Orbitrap mass spectrometer, which gives us all of the peptides. And what you can see here then is that you can see that the labeled peptide will give you a 5 Dalton shift. And you can quantify that, and you can also, because it's an Orbitrap mass spectrometer, you can identify the cysteine residues by sequencing the peptide. And we get 4 cysteines here on the 75 kilodalton, on the ND3, and on the B8 subunit. So which of those might be the one that they're important? Well, remember that complex 1 was only susceptible to modification during the inactive state of the complex 1. So what we can do is we can see in vitro, is complex 1 modified? And you can see that it's S-nitrosated on all of these in vitro. But if we take complex 1 and make it inactive, mimicking the inactive state that's present in the heart, then in that situation, we'll make it active. So it's no longer S-nitrosated on this one, but the other ones don't care. So in other words, this seems to be the candidate, the ND3 cysteine 39, because it's modified when it's inactive, but it's not modified when it's active, okay? Just by giving substrate to complex 1. The others are modified all the time, they don't care. So can we extend this to the heart? I'll skip that. Can we extend this to the heart? Yes, what we can do here is we can show that in the anoxic heart, these cysteine residues, they're all modified here equally between these. It's just 10 minutes left on this. That's modified equally here, but for this the ND3, it's not modified in the normoxic heart, but it's modified extensively under ischemia. In the mouse, the same sort of thing. If you look at ND3, what you can see is extensively modified under the ischemic heart, but not under the normoxic heart. And when you do this modification, it's rapidly reversed by thiols inside the mitochondria, both in vivo and in the mitochondria, with a half-life of about 5 minutes. So all of this is consistent with the ND3 cysteine being responsible for decreasing the activity of complex 1, and the ND3 cysteine is only available during ischemia and then is reversed later on by thiols. So where is the ND3 cysteine in complex 1? Well, we know that if we model the complex 1 here, this is how complex 1 looks. We've got this is the mammalian complex 1 modeled here based on the thermos structure that Leo Sazonov did in our unit in Cambridge. We have the flavin here. Electrons come in, bounce down the iron sulfur centers, come to the Q binding site here, and there's a conformational shift that pumps protons through conformational changes here. The ND3 cysteine just sits right here at this apex here, near the Q site where it picks up electrons, and the cysteine is there. So it's at a critical site, and we know from other work that this cysteine is exposed during anoxia from work by Salvador Moncada's lab by Alex Golkin. And so this cysteine fits in exactly with what we'd expect, and it's in an area where if it's modified, it should affect activity. Here's a closer look where you can model the cysteine to be right here, right near one of the protein translocation movements here, close to the Q binding site. So we think there's activity around this site here as the complex moves back and forth. So this is the model of what we think is happening. We have normal complex 1, NADH comes through and it does its thing. During ischemia, what happens is complex 1 goes to an inactive state, the cysteine on ND3 is exposed, and it goes into a deactive transition. But upon reperfusion, this rapidly goes back into the active form and produces a burst of free radicals just here. If we have MitoSNO present, it comes in and now it can modify this thiol here with S-nitrosating it, slowing down the activity of complex 1, preventing this burst of radical production during reperfusion. So you inhibit the ROS production, and then this is slowly regenerated by thyrodoxin and glutathione in mitochondria with a half-life of 5 minutes. And over that 5 minutes or so, you've washed out many of the metabolites that would cause the ROS production, and therefore you've critically slowed down the activity of complex 1 during this key few minutes of reperfusion. Can we generalize that? Well, other modifications that are cardioprotective also modify ND3, like nitrite is known to be cardioprotective, and again we can see extensive modification of nitrite, but only during ischemia when the ND3 cysteine is around, not when it's not anoxic. And also ischemic preconditioning, again you get extensive nitrosation of the ND3. So this suggests the following model, that during ischemic refusion injury, you get superoxide from complex 1, produce hydrogen peroxide, together with calcium, induce the mitochondrial permeability transition pore. But if you can block the reactivation of complex 1 transiently during reperfusion with MitoSNO or nitrite or ischemic preconditioning, then you prevent this point here. And this is just a cartoon to illustrate this. This is complex 1, which in structure looks a little bit like a steam engine, and we can see that it produces lots of ROS during ischemic refusion injury, but nitric oxide can hang onto the brake of the steam engine, protecting the poor heart on the tracks here. So those who actually did the work, very good PhD student and now postdoc, Edward Shoshani, drove most of this project, and it was in collaboration with the lab of Thomas Krieg and also with Paul Brooks, who did a lot of the cardiac work here. And the MitoB work to measure hydrogen peroxide was done in collaboration with Helena, Linda, and Richard in Glasgow. So thank you very much.
Thank you very much, Mike. This exciting talk is now open for discussion.
I was wondering whether you had a chance to measure oxygen consumption in isolated mitochondria treated with MitoSNO. After all, nitric oxide is known to inhibit mitochondrial respiration. So I was wondering the concentration that you use to avoid this?
Yeah, we're using a very, very low concentration. If you go to higher concentrations, then you end up with a build-up of free NO, and that competes with oxygen at cytochrome oxidase, but only under hypoxic conditions. That doesn't apply here, where the low conditions are only affecting complex 1.
Thank you for this remarkable presentation. Have you ever looked at brain mitochondria? Because what happens in vivo with cardiac arrest is that the brain suffers actually worse than the heart. And when you reperfuse, you have all these events occurring, and cerebral apoptosis is probably worse a problem than is the cardiac. So have you looked at the brain?
We are looking at the brain in collaboration. We're not looking at the brain during a cardiac event, we're looking at the brain during a stroke model, where the processes are probably very similar. And the results are very preliminary, but it suggests that similar mechanisms and similar protective strategies can be used in the brain. But this is where we're looking directly at the brain during a stroke model. We haven't looked at the brain during a heart attack model, which would be very good to look at as well.